Hi, it's Dr. Bard. I love me some histology. What's our topic today? Hello, and welcome to the wonderful world of histology. Yay, histology, go! So most of my images are from these sources here, textbooks, atlases, websites. I'm not sure if there's Enkis floating around that you can use, but definitely if you do want to start some Enki of just visual recognition, that would be a great start. I am also trying to create my own, Nordicom histology image bank. So if you are interested, hit me up. What is histology? It's the study of microscopic structure and composition of biologic tissues, which is a branch of anatomy called microscopic anatomy or microanatomy. It can also overlap with pathology. You may come across the term histopathology, where we're studying the microscopic detail and identification of diseased tissue. In the structure and function course, we're going to evaluate the normal histology of four major tissue types. We're going to start off with nervous system, and then we will cover muscle, connective tissue, and epithelium. Recall from cell biology, the cell is outlined by a plasma cell membrane that keeps everything in. Within the plasma membrane, you have hydrophobic, hydrophilic phospholipid bilayer, cholesterol proteins, glycoproteins, and the role of these, the cell membrane is to have some type of communication with the outside world. It can have receptors, it contains everything within the cell, even electrolytes, and all the juices inside the cell cytoplasm. Within the cell contents, the cytoplasm can contain various organelles, like endoplasmic reticulum. If it has ribosomes attached to it, it's called the rough RER. If it's without, it's called the smooth RER. You can also have Golgi body apparatus, mitochondria, lysosomes, vacuoles, etc. Within the nucleus, the main thing that's in there is your chromatin and nucleolus, and also don't forget the nucleus is surrounded by a nuclear membrane. So from the previous slide, we understand what a single cell looks like. Histology actually explains the visualization and the physical features of cell shapes. We're actually not going to see figures, but we're actually going to see the actual cell on light microscopy or ultrastructure. We can also see its cell compositions, and specifically these organelles on ultrastructure, and then also, and even nucleus and cytoplasm on light microscopy. We also want to see how the multiple cells are connected to each other and that how they're arranged into various tissue arrangements. Histology integrates with physiology regarding its cell function and what properties it can do, like secreting and making certain hormones, etc., or proteins and enzymes. And so you will be seeing some integration of histology with physiology in this course. And in this course in particular, I want you to really look at the visual aspects of histologic cell features. It's very important to know the normal now, because when we get into pathology in the second year, we need to be able to know, okay, that does not look normal. And so we need to be able to recognize that that cell has gone into an abnormal disease state. Four essential properties. Cells need to produce and renew, so we're going to be talking about certain stem cells and their specific cell differentiation, and if they are capable of differentiation. Certain cells and tissues we need to identify, so what is their cell or tissue type? What do they function regarding their cellular and tissue structure? Cell adhesion, we're going to talk about cells that are how they're connected to each other, and then how cells communicate with each other within a tissue, because they just don't ignore each other. Some cells do, but most cells will communicate. Yes, you have introverted cells as well as extroverted cells. Time you get your digital images, how do we get to that point? How do we obtain tissue samples? Tissues are removed and then they will be sectioned. Then they will be processed and embedded into paraffin. Once they are embedded into paraffin, we will section those tissues and transfer them onto glass slides, where we will stain them to help further identify the cell or tissue type. So we will review the glass slides in the laboratory, and in the lectures, you'll be seeing various digital images in two major forms: light microscopy or transmission electron microscopy. Light microscopy is where light is passed through tissue samples, creating an image. Tissue staining, and the purpose of tissue staining is to identify tissues, outline tissues and cell components, and identify normal or disease states. The main H&E or hematoxylin and eosin stain is a standard stain. Hematoxylin can stain nuclei, chromatin, ribosomes deep blue, basophilic, blue is basophilic, or eosin stains for cytoplasm, collagen, or connective tissue pink, acidophilic. Aside from H&E staining, you can have cytochemical stains, where cytochemical reactions occur on the glass slide with certain dyes to identify certain structures, molecules, and organisms. Examples of some commonly used stains that can help identify microorganisms are your Gram stain, your AFB for TB, Ziehl-Neelsen stain, or your India ink for a fungus called Cryptococcus, particularly in the cerebrospinal fluid. Red blood cell smears, like a peripheral blood smear, will be stained with a Wright stain or a Wright-Giemsa stain. And then proteins can be highlighted by PAS stain, and other stains such as silver stain can highlight basement membrane and other microorganisms like fungus, an H. pylori bacterium, or iron stain can help identify iron deposits. Under special stains, you also have immunohistochemical stains. Recall this concept from Dr. Romana's lecture. So I need you to bring old material to current material. We're building, we're boards reviewing, and we're adding to our knowledge bank. The immunohistochemical stains are used to detect presence of specific protein markers using specific immunohistochemical dyes. So this is where we would actually have a dye labeled antibody that will be tagging the certain protein. So antibody will recognize antigen on the cell surface, of the tissue stains, and it'll stain some type of brown or pink stain. So this is used to detect specific protein markers and assist in cell classification or tissue diagnosis. Stains, we also have immunofluorescent staining, IF stain. This stain will come into play when we start to discuss microbiology, immunology, and pathology starting next semester and next year. This identifies and visualizes distribution of proteins and other molecules in cells. It uses fluorescent dyes tagged to antibodies, which will bind to antigens in the sample. And so the images on the bottom here, you see are examples of the syphilis organism on the left, and then the skin is a pemphigus vulgaris, which fluorescent stained antibody tags are highlighting and targeting the specific proteins around the keratinocytes. Microscopy is called transmission electron microscopy, TEM, or generally termed EM. It uses high energy electrons transmitted through solid structures in order to visualize normal and abnormal cell ultrastructure. Ultrastructure is the key. The image on the left here, we see actually we have glands with a nucleus. The ribbon-like structures are RER, and the dark brown to gray structures there are your secretory granules. On top right, you have a muscle fibers containing your muscle actin and myosin and your contraction bands. You have also on the right bottom here, we have bronchial epithelium, which contains your cilia. Can you see all these little projections there? Those are your bronchial cilia. And visualize. So the difference in light versus electron microscopy is that light microscopy, you get the overall features of the cell and just the cell organization or shape. You can see the nucleus, the nucleolus, the cytoplasm, and the outline of the cell membrane. Transmission electron microscopy allows you to see high resolution details of specific organelle structures, proteins, and other cell properties. Here with the recap of this lecture. On that note, contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye!